Hi, I'm Winnie.
And I'm Alice.
And welcome to Winnie Diao
And Alice Ouyang
presents
Keeping Up with the IB Biologists. Today we're going to learn about cell structure
and functions. So as we're talking about the structures and functions of cell organelles today, the most important organelle within cells is the nucleus. The nucleus is a membrane-bound organelle found in eukaryotic cells, which contains the genetic material of the cell.
But Winnie, I heard some cells don't have a nucleus.
Yes, you are right. Those cells are called prokaryotic cells.
Do they have DNA then?
Oh, of course! Their DNA is called circular DNA, and they're contained in the nucleoids.
You're so smart.
I know, right? Okay, so the nucleus contains the chromosomes consisting of DNA associated with histone proteins. The nucleus is where DNA is replicated and transcribed to form mRNA, which is exported via the nuclear pores to the cytoplasm. And the structures of the nucleus. First, there is a thing called the nuclear envelope. So the nucleus is surrounded by a double membrane called the nuclear envelope, which separates the contents of the nucleus from the rest of the cell. And the nuclear envelope is perforated by nuclear pores that regulate the exchange of molecules between the nucleus and the cytoplasm. And second is the nucleoplasm. The nucleoplasm is a gel-like substance that fills the nucleus and contains the chromatin, which is the genetic material of the cell. The chromatin consists of DNA and proteins, which together form chromosomes during the cell division. And third, it's the nucleolus. The nucleolus is a structure within the nucleus that is responsible for the production of ribosomes. The nucleolus is composed of ribosomal RNA, which is called rRNA, and proteins. And fourth is Chromosomes are the condensed form of chromatin that is visible during cell division, and they contain the genetic information of the cell in the form of DNA. And that's all about the structures of the nucleus. Well, of course, not all the structures, but that's enough for you to learn biology.
And get a 7.
Yes, you are right. Okay, so now we're moving on to the functions of the nucleus. First, the most important function of the nucleus is genetic information storage. And the nucleus houses the cell's genetic material in the form of chromosomes. It contains DNA, which is short for deoxyribose nucleic acid, which carries the instructions of the cell's development, growth, and functioning. The DNA is organized into structures called chromosomes, which consist of genes that encode specific traits and characteristics. And second is gene expression and regulation. The nucleus plays a crucial role in the gene expression and regulation. It contains the transcriptional machinery, including RNA polymerase enzymes and transcription factors, which control the process of converting DNA into RNA. This RNA can be further processed and transported out of the nucleus for protein synthesis, which is translation, or can have other regulatory roles within the cell. And third is DNA replication. So the nucleus is responsible for DNA replication, the process by which DNA molecules are duplicated to ensure the accurate transmission of genetic information to daughter cells during cell division. DNA replication occurs during the cell cycle to ensure that each daughter cell receives a complete set of genetic material. And that's all about the nucleus, which is like the center of the cell.
That's so cool.
I know, right? And now we're moving on to another organelle, which is also very important, and it is called the plasma membrane, or known as the cell membrane, but now in IBDP we call it the plasma membrane. So the plasma membrane, also known as the cell membrane or the cytoplasmic membrane, is a thin, flexible barrier that separates the interior of a cell from its external environment, and it is found in all types of cells, including prokaryotic and eukaryotic cells, and plays a critical role in regulating the movement of molecules in and out of the cell. And there are a few structures that you need to know about the plasma membrane. First is the phospholipid bilayer. The plasma membrane is composed of a double layer of phospholipid molecules with their hydrophilic heads facing outward. Hydrophilic, by the way, means water-loving. Their hydrophilic heads facing outward and their hydrophobic, which is water-fearing, tails facing inward. So this creates a hydrophobic barrier that prevents the movement of most hydrophilic substances across the membrane. And second is called the membrane proteins. So embedded within the phospholipid bilayer are various types of membrane proteins, which plays a variety of roles in regulating the movement of molecules across the membrane. Some proteins act as channels or transporters, so allowing specific molecules to move in and out of the cell, while others act as receptors binding to specific molecules and triggering cellular responses. And third, is the carbohydrates. So many plasma membranes also contain carbohydrates, which are attached to proteins or lipids, and which helps to organize and interact with other cells and molecules in the cell's environment. And then the last one you should know is the cholesterol. So in animal cells, the plasma membrane also contains cholesterol molecules, which help regulate the fluidity and stability of the membrane. And remember, cholesterol only exists in animal cells' plasma membranes. And that's all about the plasma membrane. Okay, and then we are moving on to the cell wall. Oh my god, this is so cringe. Okay, the cell walls are rigid protective structures that surround the plasma membrane of many types of cell, including...
Oh, do I have a cell wall?
No, of course you don't, because cell wall only exists in plant cells. I mean, in plant cells, bacteria cells, or fungi, and of course some protists.
That's not fair. I want a cell wall.
Okay, fine. So they're rigid, protective, and surrounded the plasma membrane of many types of cells, but not animal cells. Okay, so the structure of cell walls can vary depending on the type of organism. So in plant cells, the cell wall is composed primarily of cellulose, which is a carbohydrate polymer. And in bacteria, the cell wall is made up of peptidoglycan, which is a complex polymer made up of sugars and amino acids. And fungal cell walls are composed of chitin, which is a nitrogen-containing polysaccharide. And the functions of the cell wall, so it provides structural support for the cell. So the cell walls help maintain the shape and the integrity of the cell, which is particularly important in organisms that do not have a skeleton or other internal support structure. And the second function is to regulate the water balance. Cell walls can help regulate the movement of water in and out of the cell, which is important for maintaining turgor pressure and preventing dehydration. And the last one is protecting the cell against pathogens. So many cell walls have properties that make them resistant to the invasion by bacteria, viruses, and maybe some kind of other pathogens, and which provides an additional layer of defense against infection. And that's all about cell wall.
Now I'm going to tell you all about the cytosol.
Hey, smart biologist, what is cytosol? It sounds like cytoplasm. Are they the same?
Well, no. The cytosol is the fluid part of the cytoplasm. It's also known as the cytoplasmic liquid. The cytoplasm is everything within the cell membrane, but the cytosol is the part that is fluid. So the cytosol has several important functions. First of all, it's a site for a series of important metabolic functions. Many enzymatic reactions involve cellular metabolism, such as glycolysis, and they all occur within the cytosol. Also, the cytosol is very important because of its role in storage and transport. So particles are in a liquid state. They can flow around more easily. So that means they can collide with each other more easily. And according to collision theory in chemistry, this would facilitate the metabolism going on in the cells. And another important part of the cytosol's function is its storage and transport. The cytosol acts as a storage site for various substances, including ions, metabolites, and small molecules. It also facilitates the transport of these molecules within the cell, providing a medium for diffusion and molecular movement. And that's basically the function of the cytosol. You just have to remember it's the fluid part of the cytoplasm. Now we are moving on to ribosomes. Ribosomes are like the little factories of the cell. They are the manufacturing center for proteins. They carry out protein synthesis. Now, ribosomes are made of ribosomal RNAs and proteins. There are two types of ribosomes, free ribosomes and bound ribosomes. Bound ribosomes and free ribosomes are structurally identical, but they perform different functions. So bound ribosomes manufacture proteins that are destined for insertion into membranes, for packaging within certain organelles such as lysosomes, or for transport of excretion out of the cell. And that's it for ribosomes.
So now we're moving on to the mitochondria. The mitochondria are double membrane bound organelles found in most eukaryotic cells.
Oh, Winnie.
What?
I heard that the mitochondria are called the powerhouse of the cell. Why
Actually,
is that?
you are right. So mitochondria are often referred to as the powerhouse of the cell because their primary function is to generate energy in the form of adenosine triphosphate, which is also called and also known as the ATP. And they generate ATP through a process called the cellular respiration. And about the structures of the mitochondria. So the structure of mitochondria consists of an outer membrane that encloses the entire organelle and an inner membrane which is highly folded to form structures called cristae, spelled as CRISTAE. And the inner membrane separates the mitochondria into two compartments: the intermembrane space and also the mitochondrial matrix. And about the functions of mitochondria, as I just mentioned, the primary function of the mitochondria is to release ATP through cellular respiration. But in addition to that, mitochondria are also involved in other cellular processes, such as regulating cell death and calcium storage and the synthesis of certain molecules. And they also contain their own DNA, also known as the mitochondrial DNA or mDNA, and which encodes a small number of genes necessary for mitochondrial function. And next, we are moving on to the chloroplasts. Chloroplasts are specialized organelles found in plant cells and some protists that are responsible for
photosynthesis. What
Yes.
is photosynthesis anyways?
So photosynthesis is the process by which plants, only plants, and some other, but not animals, convert sunlight into chemical energy. Most of the animals don't have chloroplasts, okay? Because they eat, they don't synthesize.
All the cool kids have chloroplasts.
Whatever. Okay. So, chloroplasts are responsible for photosynthesis, which is the process by which plants convert sunlight into chemical energy. And the chloroplasts contain a green pigment called the chlorophyll with 2 L's in the end, which gives plants their characteristic green color.
That's so pretty.
I know, right? And about the structures of the chloroplasts, the structure of the chloroplast consists of an outer membrane and an inner membrane, with a space in between known as the intermembrane space. And inside the inner membrane is a substance called the stroma, which contains enzymes, DNA, and ribosomes. And its functions, as always, is to carry out photosynthesis. And that's all about the chloroplasts.
And now, kids, it's time to learn about the smooth and the rough ER. So, the smooth and rough ER are actually called the smooth and rough endoplasmic reticulum. Long, fancy name. They are called smooth and rough because the rough ER has ribosomes embedded on the outside and the smooth ER does not. So the primary function of the rough ER would be protein synthesis. But it's not actually the rough ER that does the protein synthesis. It's because it's studded with ribosomes, which are responsible for synthesizing proteins, and it plays a crucial role in the synthesis of membrane and excretory proteins. Most excretory proteins are glycoproteins. The rough ER is also very important in protein folding and modifications. As proteins are synthesized, the rough ER provides an environment for proper folding and modification, such as glycosidification, disulfide bond formation, ensuring their functional conformation. And the rough ER is also important for quality control. So the RER participates in quality control mechanisms by monitoring protein folding. Now, misfolded proteins or improperly assembled proteins are recognized and targeted for degradation or further modification. Now, that's very cool. And finally, the RER is also a kind of highway for molecules, so it's very important in intracellular transport. Secretory proteins depart from the ER wrapped in the membrane vesicles that bud like bubbles out of a special region called the transitional ER. Now, these vesicles transit from one part of the cell to another part of the cell, and they are called transport vesicles, very aptly named. Now, moving on to the smooth ER. The smooth ER has slightly different functions. So instead of participating in protein synthesis, it carries out lipid metabolism. So the SER is involved in the synthesis of lipids, including phospholipids, so our good old cell membranes, cholesterol, and steroid hormones. It also participates in the breakdown of fatty acids and the detoxification of lipid-soluble drugs and toxins. The SER is also important in calcium storage and release. So it serves as a reservoir for calcium ions in the cell, and it plays a crucial role in regulating calcium levels and releasing calcium when needed for various cellular processes, such as muscle contraction and signaling. Finally, the SER is also important in glycogen metabolism. So in some cell types, the SER participates in glycogen metabolism by storing and releasing glucose when it is needed for energy production. And that's about it. Now, we are moving on to the Golgi apparatus. The Golgi apparatus is composed of flattened membrane sacs called cisternae, and besides existing in the Golgi apparatus, cisternae also exist in the rough ER. Now, it's debated whether the smoothie R has cystine A because different textbooks disagree. But the smoothie R has a slightly different structure, so I'm just going to say here that rough ER has cystine A and smooth ER does not. Now, back to the Golgi apparatus, each cystine within the Golgi stack has a distinct structural directionality with a cis face and a trans face, like the same cis and trans as cis fats and trans fats. Now, the cis phase would be the receiving department, and the trans phase would be the shipping department. So that links to the function of the Golgi apparatus, which is basically just this little post office. So it packages proteins and molecules, and it modifies these proteins and sends them out. So vesicles are concentrated near the Golgi apparatus and participate in the transfer of materials between the Golgi and other cellular structures. The Golgi apparatus, sometimes it might be difficult to discern between the Golgi apparatus and the rough ER in diagrams. So a little exam tip, I guess. When you're looking at the Golgi apparatus, you'll notice that there are little sacs bulging out on each side that kind of look like this. And you won't see these little sacs at either side for the endoplasmic reticulum. Now, back to the function of the Golgi apparatus. So the Golgi apparatus is responsible for receiving, sorting, and shipping. The Golgi apparatus acts as a warehouse for receiving, storing, modifying, and shipping various products, including proteins and lipids. It's also responsible for modification, which I mentioned earlier. So products from the endoplasmic reticulum, such as proteins, undergo modification in the Golgi apparatus. This includes the modification of carbohydrates attached to the proteins, so glycoproteins, and the alteration of membrane phospholipids. Now, finally, the Golgi apparatus is important for vesicle formation and transport. So vesicles bud off from the Golgi apparatus, carrying specific products to other cellular locations, to the plasma membrane for secretion, and some vesicles also transport proteins back to the ER for their functional sites. So overall, the Golgi apparatus plays a crucial role in receiving, modifying, sorting, and shipping various cellular products, ensuring their proper distribution within the cell and secretion to external destinations. And that's it. Yay!
Okay, so thankfully we are almost at the end of this very first video, and now we're moving on to lysosomes. So lysosomes are membrane-bound organelles found in animal cells, and they're often referred to as the cell's recycling centers or digestive organelles. This is because their role in breaking down waste materials and cellular debris. So lysosomes contain a variety of enzymes that are responsible for hydrolyzing, which is breaking down with water, various molecules. And about the structures of the lysosomes, they're formed by Golgi apparatus or Golgi complex or Golgi body, whatever. So, which, as Alice mentioned, packages enzymes into vesicles, and these vesicles then fuse with endosomes, creating mature lysosomes. Lysosomes have a single membrane that helps to contain the digestive enzymes within the organelle. The interior of a lysosome is acidic, maintained by proton pumps on the lysosome membrane that pump hydrogen ions into the lysosome, creating an optimal pH for the enzymatic activity. About the functions of lysosomes. So first is the intracellular digestion. Like I just said, it's referred to as the digestive center. So lysosomes contain enzymes that break down various biomolecules, such as proteins, lipids, carbohydrates, and nucleic acid. And they help in the digestion of cellular waste materials, recycling them into useful components of the cell, for the cell. And the second one is waste removal. Lysosomes play a very crucial role in removing unwanted or damaged cell components, and they fuse with vesicles containing wastes or obsolete organelles, breaking them down and enabling the recycling of building blocks for new cellular structures. And the third one, which is the last one, we think that you should know and which is helpful for exams and cores and whatever, is to defend against pathogens. Lysosomes participate in the immune response by fusing with phagocytic vesicles that engulf invading pathogens or foreign particles. The enzymatic components of lysosome aid in the degradation and elimination of these pathogens, protecting the cell and the organism from infections. And we're moving on to the very last organelle of today, which is peroxisomes. So there's actually not that much to talk about peroxisomes, but they're also included in the IB content, so you should know something about it. So peroxisomes are small single membrane bound organelles found in the eukaryotic cells, and they're involved in various metabolic processes and play a crucial role in the detoxification and the breaking down of fatty acids. And its structures. Peroxisomes have the characteristics of spherical or oval shape and contain a dense protein matrix. And they're surrounded by a single membrane that separates the content of the peroxisome from the cytoplasm. Within the peroxisome, enzymes and proteins are present there that are necessary for its specific functions. Talking about its functions, well, the first one, which is also considered the most important one, is detoxification. So peroxisomes are involved in the detoxification of harmful substances within cells, and they contain enzymes such as catalase that break down toxic hydrogen peroxisomes into water and oxygen, which prevents the accumulation of reactive oxygen species that can damage cellular components. And the second one is fatty acid metabolism. So peroxisomes are crucial for the breakdown of fatty acids through a process called the beta oxidation. Enzymes within peroxisomes break down long-chain fatty acids into shorter chain forms, and which can then be transported to the mitochondria for further energy production. Well, as you might remember, mitochondria is the powerhouse of the cell, so yeah. Okay, so the last one, which is not probably not included in your test, is that biosynthesis. So peroxisomes are involved in the biosynthesis of certain important molecules, including plasmalogens, which are essential components of cell membranes. And they also contribute to the synthesis of bioacids, which aids in digestions, which we expect you to know that, and absorb of dietary fats. And that is all of our content for our very first video. Thank you for watching. Bye.